you very much, Hugo, for introducing me so kindly. I'm very happy to give a talk today about phenotyping of plant diseases. And most of my talk will concentrate on using hyperspectral imaging for assessing plant diseases on different scales. However, I will start a little bit more generous. So, how has agriculture developed during the last 100 or nearly 100 years? If we look back in time, in 1950, one farmer was able to provide food for about 10 people, and it was quite hard to cultivate the land. Then there was a huge innovation, mainly to technical progress, and one farmer in 2013 was able to provide food for nearly 150 people. And now the interesting question is, how will it be in the future, 2050? Arable land is getting less, and we have to be much more effective in the future. And I think the hypothesis is that this can be done by technological process and better organization between input data. And here data and sensing and the overall scope of PhenoRob is really crucial. So what are the two main application areas of assessing plant diseases? One application area is precision agriculture. And if we have a look into the field, plant diseases often exhibit spatial heterogeneity within crop stems. And this is due to different temporal appearance depending on external factors like temperature, humidity, the inoculum density within the field. And therefore, we do not have the plant diseases on the same disease severity level on each area or on each part of the field. It's more this kind of patchy distribution. And now it would be interesting to use different kinds of sensors combined with geoinformation systems to assess these patches of the diseases and to combine it with expert systems or information about the environment to define a management practice which can be used to reduce the application of plant protection measures and which can help the farmer to guide his actions. The second main application is breeding and phenotyping, but here the situation is totally different. We have much more controlled conditions. The phenotyping pipeline or the breeding pipeline is roughly demonstrated here, where we start with a huge number of genotypes, and the aim of the breeder is to define the best performing variety to launch it into the market. And there are some aspects which are very time consuming. It's the lab and greenhouse phenotyping and field phenotyping. And here we have a huge potential to accelerate these processes by non-invasive sensors. However, as I mentioned, the situation is here much more controlled compared to the field application of the farmer. We have a field plot design. Often we have sometimes an artificial inoculation of the pathogens. We know which pathogen should appear in the situation, and here it's more about resulting or creating and ranking of the genotypes to select the best performing genotypes. How can we use hyperspectral imaging, therefore, and what is the theory behind? The way of light is briefly demonstrated here. We have the leaf surface with the different structures, the cuticular, epidermis, palisade, parenchyma, and when incoming light hits on the plant leaf, it goes different ways. Some part of the light is transmitted through the leaf, some parts of the light is absorbed depending on internal factors, the plant biochemistry, the water content, pigment content, etc. And what we are able to measure with our hyperspectral sensors as an information from the plant is composited by internal reflection and the direct reflectance on the upper surface. So this is the normal situation when a plant leaf is quite healthy and well established. However, if we have an interaction with plant pathogens, there are several structural changes and also changes in the biochemistry. You see here 3 examples of plant diseases with a different lifestyle. 2 biotrophic pathogens, powdery mildew and leaf rust, and 1 pathotrophic pathogen, a leaf spot disease. And you see here in color the different fungal structures and how they interact with the plant tissue. Powdery mildews, for example, parasitize especially the epidermal cell layer, and they develop a mycelium on their upper leaf surface with cornelia, etc. Rust diseases grow in between the palisade parenchyma cells and produce these kind of haustoria to feed from the plant tissue. And pathotrophic pathogens like leaf spot diseases produce toxins to destroy the cell tissue and feed from that cell tissue and therefore have the highest impact on the plant structure. And if we have a look back now on this slide and on the way of light through the tissue, you can imagine that these kind of specific interactions of the plant with the pathogen influence the way of light in a characteristic way, and this results in characteristic spectral reflectance patterns. So this is the theory behind what kind of data do we obtain by hyperspectral imaging. We obtain these kind of hyperspectral data cubes, which are shown here on the left side. And these are three-dimensional images. We have the two spatial dimensions, x and y. And additionally, for each pixel, the spectral dimension z. And this is shown here. We obtain for each image pixel a spectral signature in a specific wavelength region. And here it was from 400 nanometers to 1000 nanometers. And depending on the situation and the health status and the vitality of the tissue, these spectral signatures are influenced in a specific way. For example, you see here in green, the dark green line healthy parts of the leaf from the left side, and in red, a highly diseased part, and you see that in the visible range, we have changes from 400 to 500 nanometers and also from 450 to 700 nanometers, but also the near-infrared range, which is not visible for our eye, is influenced depending on the disease severity. So as you can imagine, we obtain a huge amount of data, which can quite fast grow into terabytes. And now there's the interesting question how to analyze these kind of data and how can we derive the information we need from the data in a quite fast way. And there are different kind of approaches, some quite not old school, but more conservative approaches like calculation of spectral features, so-called spectral vegetation indices, which is quite common in research, and it started from remote sensing. And here we use only a few wavelengths of the spectrum and calculate an algorithm. And this algorithm, this spectral vegetation index, is correlated to a characteristic of the plant, like chlorophyll content or anthocyan content. And here we have a data reduction, so we don't need we don't use the entire information of the spectral signature, so information is lost, but it's an easy way to get a result quite fast and with not so much calculation or automation effort. Other approaches are automatic classification approaches. These can be supervised or unsupervised. Some examples are just supervector machines, SIBIM, or K-means. But the overall aim for plant diseases is the disease detection, and here we need the detection where is the disease, as you can see it here on the sugarbeet leaf with the Cercospora. We want to identify the disease, so is it a leaf spot or is it a powder mildew or a rust disease? And of course it's very important to quantify the disease to assess the disease severity from the hyperspectral images automatically. So just some few examples on the next slide how this can be performed. These are sugarbeet leaves which were measured with a hyperspectral imaging sensor from 400 to 2000 nanometers in the visible range and in the near-infrared range. And we did an inoculation experiment, so we infected the plants with the disease and we measured the development of the leaves starting from 0 points after inoculation until disease appearance. So we have here the first day when the first spot appeared. It's a quite small spot, so plant diseases are very tricky because they start very small and only a few millimeters are influenced into the tissue. And for leaf diseases, we have the experience that there is not a huge systemic effect. So you really need to assess the spot where it starts to appear. However, we applied here a clustering approach, so it was more or less a data-driven approach, where we separated different kinds of tissue in the leaf, so the healthy tissue, the leaf veins, and on 15 days we were able to assess all leaf spots and also the different kind of structures of the leaf spots. You see here in red the highly destroyed necrotic center of the leaf spots, and in yellow the surrounding tissue, so the border between the diseased spot and the healthy tissue. And based on these images, you can just calculate the amount of disease pixels, so then you have the quantification, and based on the spectral signatures, we are able to also characterize the Cercospora leaf spot disease. When we show these images, always the first question which arises is, are you also able to assess the diseases before symptoms are visible? And we can say yes, it is possible, but only under controlled conditions. So far in the field, it's very challenging. The environmental factors are not optimal for the measurements, but if we have a highly controlled setup, we are able to differentiate different diseases like Cercospora leaf spot, Sugarbeet rust and powdery mildew by support vector machines, and we are also able to assess or to define the inoculated plants before symptoms are visible. And this is seen here, where we did a comparison between the visual monitoring by a human to the monitoring by the support vector machines based on hyperspectral data. And this was possible with an accuracy of 93%, so we were quite happy about these results. So after a few years working on assessment of diseases on plants, crop plants, we started to also investigate resistance of plants against pathogens, and resistance is also on a much smaller level. You see here the example of barley and powdery mildew. So if a plant is resistant against a pathogen, there are differing underlying resistance mechanisms. So for example, in the system barley and powdery mildew, there's a well-described resistance which is called MLO resistance, where the plant is able to produce a so-called papille. It's a structure out of lignin, and this structure inhibits the pathogen, and the pathogen cannot penetrate into the tissue, cannot develop the haustoria, and therefore the pathogen cannot feed from the plant, and the plant will be not infested by the pathogen. A different resistance mechanism is the so-called hypersensitive response, HR resistance. It's mostly in MLA, so-called MLA genotypes, and here the penetrated cell induces cell death, and by this cell death the pathogen is again not able to feed from the plant and is not able to develop relevant structures like the mycelium or the conidia. And we use this well-known system. We use different genotypes, a susceptible genotype, MLO3 genotype with the papilla as a resistance, and the MLA1 genotype with the hypersensitive response as the resistance. And we monitored these plants with a hyperspectral microscope on a very small scale to investigate whether we are able to see differences based on the genotypes. Here you see a timeline, a measurement timeline of the susceptible genotype from 0 days to 40 days after inoculation, and the resistant genotype from 0 to 40 days as well. And if we have a look on the healthy control, the wild type develops quite normal, so we start with a typical plant reflectance curve. The leaves get senescent over the experimental period, and this can be also seen in our signature. And if we compare it to the MLO3 genotype, we have a quite similar pattern over time. But interestingly, if we look now on the inoculated plants, which were infested with the powdery mildew pathogen, we see that in the wild type, the susceptible one, the disease is able to develop, and this is also reflected in the signature. And if we have a look on the resistant genotype, we see that there's nearly no change in spectral signature over time. Only at the last day, 14 days after inoculation, when the leaves are quite senescent on the agar plates, we see differences. So we have here a differentiation between the wild type and the resistant one is here visible. And what was new to us, we expected that the MLO3 inoculated should look similar like the MLO3 healthy, but what was observed here was that there was a delay in senescence. So the interaction between the pathogen and the resistant genotype resulted in a delay of senescence, a longer vitality of the resistant genotype, and this was very interesting to monitor by the hyperspectral microscope. So we also used these data to differentiate the genotypes by data mining approach. This was in collaboration with Uwe Rascher and Christian Kersting out of CropSense, and we were able to create these kind of disease maps to locate the pathogen on the susceptible genotypes, but we were also able to develop these kind of spectral traces where we could differentiate the resistant individuums, here in green, from the susceptible individuums in black. The second question was if we are also able to locate hypersensitive responses in the tissue. So the hypersensitive reaction in the tissue is also very, very small. It's only a single cell which develops these kind of reactions, the hypersensitive reaction. And we had a look by classical microscopy, DUP staining, and we developed also chromatography and view staining to see if there is a reaction or not. And we monitored the leaves again with the hyperspectral microscope, and you see that on 2 days and 3 days after inoculation, it's not possible to see hypersensitive responses by the eye. 5 days after inoculation, there are some hints that there are hypersensitive reactions on the leaves. But if we have a look now with the data mining approach, also clustering approach again, on our data, we were able to indicate or to locate these hypersensitive responses. Nevertheless, there is still some noise on the leaf, so the leaf is not heterogeneous, therefore there's still some noise, but with a subsequent thresholding approach, we are able to locate the hypersensitive responses from our barley leaves. So one last slide on assessing resistance of plants against pathogen. We change the system now, go back to Sugarbeet and the system Sugarbeet Cercospora leaf spot. Here we also had an investigation of different genotypes. There was a genotype with an QTL, which was resistant against Cercospora leaf spot, and a genotype without a QTL, which was susceptible. However, in this situation, both genotypes showed symptoms, but the response to the yield in the end was different. And we had a closer look to the symptoms, if we were able to see differences. And we investigated that the symptoms of the resistant genotypes had a quite stable appearance over time, whereas in the susceptible genotypes, there's a higher dynamic over time, and different spectra can be extracted from our hyperspectral images. So we were wondering what is going on here and did some raster electron microscopy. And with this closer look, we were able to see that the resistant genotype, there are no spores developed in the center of the spot, whereas the susceptible genotype develops spores. And the spores are important for the further distribution of the pathogen. So we have an initial infection in the resistant genotypes, but due to the low amount of spores, the disease severity during the vegetation period is much lower compared to the one where the spores develop in the center. And this can be monitored by hyperspectral imaging. So what might help the farmer now? So how can we bring all these technologies closer into the field and closer into practice? And here we have in collaboration with Christian Bauchhage and SEP and ISIS, where the idea was to develop a smartphone app for the farmer. The farmer can go into the field, take a picture, just an RGB picture, and the app should provide information to the farmer which kind of leaf disease appears on his sugar beet, and if it is a fungal disease or a bacterium, etc. And this app can be downloaded here on EASY.de or on the KWS homepage. And how was the algorithm? I think that Christian can explain it much better than me, but the principle was that the farmer goes into the field, assesses images, then a region detection algorithm was applied on the images, which identifies regions which are not healthy. These ROEs were transected or were extracted from the image and were then sent to the server where the feature extraction and classification was performed, and in the end the app is able to differentiate among 5 relevant diseases of sugar beets. And what was very important here for this setup is that the algorithm and the app is able to handle all kinds of images. The images don't have to be taken in a specific setup, so just snapshots from the farmer, and in the end it went quite well. So what are we performing or doing currently in our research? Right now we have a project on data-guided management of wheat diseases, and I think you know this slide already. The idea here is to sense the crop stand, wheat crop stands in the field, to detect images based on a machine learning approach, create distribution maps based on this information, and to provide an application plan to the farmer in the end. However, this is a quite challenging approach, and wheat is also infested by different kinds of diseases, and therefore we had an idea to to integrate information from different scales. We assessed the characteristic spectrosignatures of fungal structures like rust spores. We assessed also the symptoms under controlled condition to have characteristic signatures, and then we went on the single plant scale to have the heterogeneity based on the architecture of the plant, and finally we also assessed information from the field with the PhenoBike and based on UAV measurements. The diseases which were investigated are yellow rust, brown rust, tan spot, and septoria. So we have 2 biotrophic diseases, 2 rust diseases, and 2 pathotrophic diseases. And the characteristic spectrosignatures differ significantly if we have a look on the mature symptoms. And what is interesting for us is that we are also able to differentiate among the 2 rust diseases, so yellow rust here in yellow and brown rust in red. And also the 2 pathotrophic diseases which cause necrosis can be differentiated in different regions based on the spectral signatures. So this was quite promising in the beginning. However, these are fully developed symptoms which can be also recognized by the human eye, and therefore we assessed the spectral dynamic over time in the time series measurements, and this is just some information about brown rust. And you see that we have also a temporal dynamic in the appearance of the spectral signatures. But we were able to define so-called turning points, which are characteristics and for a disease and which are more or less time independent. Because if we go into the field, we do not know since when the disease is already on the leaf and in which developmental stage the leaf appears. And therefore, we identified these 3 turning points for each disease to be time independent, to transfer this information from the lab into the field. So, just briefly for you, the field experiment, which was performed in Campus Klein-Altendorf in 2018. It was a plot experimental design with 2 varieties, Asano and Bussard, and 10 different treatments. We also had different nitrogen application here in the situation. We had plots with fungicide and plots without fungicides, and we had plots which were inoculated. The images which were taken were hyperspectral images of the canopy with the PhenoBike. We had an imaging line scanner and filter-based camera, and we also did UAV measurements with a hyperspectral camera, and also from Jülich UAV images with a multispectral camera were performed. We monitored the experiment over the vegetation period, beginning in April. The disease assessment for ground truth data was performed in parallel to the sensor measurements. The PhenoBike measurements were performed nearly weekly, and the UAV measurements were performed depending on the availability of the sensor and depending on the environmental conditions. So have a look on the canopy data, which looks like this. It's quite a mess if you measure wheat canopy from above with a line scanner, because the line's gonna need some time to move over the area, and if there is wind, yeah, the ears and the leaves are moving, and this is one of the better images from our experiment. And you also see that there is a huge heterogeneity regarding the illumination. We have highly illuminated leaves, we have shadows, we have different angles in the view, so it's really a challenge for the algorithm. We started with an annotation of these images for a subsequent disease classification, and we defined 5 different classes: healthy parts of the leaf, the ears, the tip of the leaves, because often the tips are brown, old leaves which are already senescent, and the background. Based on this annotated data, we extracted the canopy reflectance data, and by a first look, it's not possible to differentiate clearly the healthy leaves and the diseased parts of the leaves in blue and in yellow, because we really have this huge heterogeneity and this differing reflectance situation in each image. However, we use this data as an input data for supervised classification, and we decided to use at the beginning 2 classifiers: the spectral angle mapper, which is well established in remote sensing and support vector machines, and we compared the results of both classifiers. And these are the results. So yeah, it looks a little bit messy, but we were quite happy nevertheless in the end. In green you see healthy parts of the leaf, and in yellow you see the yellow rust parts of the leaf. And we see that the spectral angle mapper is not so sensitive regarding senescent part. However, it is highly sensitive for yellow rust. But there is a high number of misclassification regarding upper leaves and lower leaves, and also upper leaves and the ears. The ears here are often classified as diseases. The support vector machine was also highly sensitive for yellow rust, but here often a misclassification of the leaf border appeared. So this kind of approaches still have to be improved. Nevertheless, the classification accuracy was 81% for the spectral angle mapper and 92% for the support vector machine. Here also the question is, how can we get into practical use? Because we experienced in the last vegetation period that the measuring setup is very time consuming, and it's really not practical for a farmer. And therefore the idea was to perform feature extraction. So do we really need the entire information from hyperspectral data, or can we use simply multispectral data? What wavebands are necessary and what number of wavebands are necessary? And the idea was to define a sensor which is characteristic for the wheat diseases. Here are some very early results. We assessed the number of features which is relevant for a high accuracy in the classification, and it's about 12 features which are relevant. And we also performed a wrapper approach for assessing the features and the wavebands which are relevant for each disease. And here for yellow rust, we need about 10 wavebands which are distributed over the entire spectrum. So it's not a specific wave range which is characteristic for yellow rust. If we want a high accuracy, we need wavebands from the visible range and also from the near-infrared range. And we did these for all diseases, and hopefully this will help to differentiate among the diseases. Nevertheless, disease detection is quite challenging, and you can see this here. If we have the idea to use drone data for disease detection, this is a Sugarbeet field in May last year, where we can easily differentiate between plants and soil. We can detect plants, we can detect areas where no plants appear, so the biomass development can be monitored quite well. We also can detect the leaves of the Sugarbeet. This is possible. But if we want to use these kind of data to assess Cercospora leaf spots, which are about 2 millimeters or 3 millimeters, we need a different approach. We really have to get closer to our plant to be sensitive. And we tried to model or to simulate this here based on hyperspectral data, what we have. Here is a close-up view of about ½ a meter on a sugarbeet leaf. You can easily differentiate between the diseased parts and the healthy parts. And depending on the distance, the amount of mixed pixel is increasing in the hyperspectral images, and by this, the detection accuracy is also decreasing. So this is still a challenge we want to address within PhenoRob, and as a collaboration between Core Project 2 and Core Project 1, this is really an important issue which should be addressed. A second important issue which should be addressed, and you have seen it on the wheat data, is how to handle and how to deal with the plant architecture in the hyperspectral images. And let's see if it is running. This was a collaboration with a group of Anne-Katrin, where we assessed the architecture of sugarbeet plants, and in parallel we assessed hyperspectral images, and we matched the hyperspectral information, the 3D information, to develop the so-called hyperspectral 3D model. And this may help to calibrate our plant models depending on the distance between the sensor and the plant, depending on the illumination intensity, and also depending on the leaf angle, which also influences the hyperspectral signal. Yeah, with this, I'm at the end of my talk. I think we still need some key features for a successful implementation of digital technologies in agriculture, precision agriculture, and plant phenotyping, and especially for disease assessment. We do have to develop problem-orientated measuring setups, and we need a perfect timing when we go out in the field to measure. So it is crucial to characterize the best type of the sensor in combination with the platform. On top of this, we need an efficient data analysis, and we still have some hope that scale-independent approaches can be developed to increase the accuracy and the specificity of our measurements. Yeah, but in the end, we need an integration with the agriculture expertise, and as you can see that there are different challenges in different areas. We really do need the interdisciplinary approach to get the results what we need. Yeah, and this I would like to thank you for your attention, and questions are welcome.